the effect of naltrexone bupropion on major adverse cardiovascular events in overweight and obese patients with cardiovascular risk factors. This study was published in JAMA Network in March of 2016. So the objective of this study was to determine whether the combination of naltrexone and bupropion increases major adverse cardiovascular events, also referred to as MACE, compared to placebo in overweight and obese patients. And what they included in MACE was cardiovascular death, non-fatal stroke, and non-fatal myocardial infarction. So just to get started with a question here, probably more geared towards the first years, but what type of outcome is MACE in this study? And you can select all that apply.
Any other responses?
I don't know how to show the responses. I don't know if I have that capability on my end.
I will do it. I'll
Okay.
end the poll and it should view, we should see it just a moment.
Okay. Great, thanks. Great, so most people got them all right. So dichotomy, so it was number 2, number 3, and number 5 were the correct answers. So great job, everyone, and thanks for participating. Just gonna do, okay. So in terms of a bit of background on the study, there have been few cardiovascular outcomes trials that have been conducted for obesity treatments. In 1997, 2 weight loss drugs were removed from the market due to evidence of cardiovascular harm. And this really raised concerns about the cardiovascular safety of new drugs that were being created to treat obesity. So in 2008, the FDA issued guidance indicating that new anti-diabetic medications should actually be evaluated for their potential to increase cardiovascular risk. And this led to more focus on assessing cardiovascular safety in these medications. And this guidance really led to a 2-step approach to evaluating long-term cardiovascular safety profile of medications that was first proposed and applied in anti-diabetic medication studies. And so this strategy has since been implemented by the FDA to ensure that these drugs don't pose any cardiovascular risks. So we thought we would review the what might be included in the pre-approval studies, or the pre-approval phase, and then the post-approval phase. So pre-approval phase studies are typically short-term studies. So they're implemented before the medication is approved, and they're really geared towards assessing the medication's effectiveness and safety in the short term. For anti-diabetic drugs, these studies typically focus on efficacy endpoints like hemoglobin A1C or other relevant biomarkers. And then during these short-term studies, safety is continuously monitored throughout to identify and record any adverse effects or side effects related to the medication. The problem with these studies, though, is that typically they're not long enough or powered to definitively assess rare safety events and events that might occur over a longer period of time. And so that's where the post-approval studies come in. So these are typically conducted after regulatory approval, and they really aim to gather more data about the medication safety profile over an extended period of time, allowing for the detection of rare or long-term adverse events that might have not been picked up in the short-term studies. There's also post-marketing surveillance. So relevant stakeholders also engage in a monitoring process to assess the safety of a medication once it's become available to the broader population. So there was a lot of work that led to initiation of the LIGHT trial, which is the study that we're presenting today. This was the study that preceded the LIGHT trial. So in 2010, a multi-center randomized double-blind placebo-controlled phase 3 clinical trial called the Contrave Obesity Research 1 study was published in the Lancet. And the objective of this study was really to assess the effect of the combined naltrexone and bupropion on body weight in overweight and obese participants. And though they found that the combination was successful in reducing weight, the FDA deferred the approval based on safety concerns related to a small number of patients who had small increases in blood pressure and heart rate throughout the trial. And so this is just a screenshot of the Medscape article that came out at the time. So on February 1st, 2011, the FDA deferred the approval of Contrave, the combination of the two, asking its manufacturer to conduct a study of sufficient size and duration to examine the potential for cardiovascular risks. And so this letter specifically stated that before your application can be approved, you must conduct a randomized double-blind placebo-controlled trial of sufficient size and duration to demonstrate that the major adverse cardiovascular risks essentially do not adversely affect the drug's benefit and risk profile. So really mandating the LIGHT trial, which came next. And so these were the specifications of the LIGHT trial, that an interim analysis would be conducted after 25% of expected events had accrued to rule out an upper 95% confidence interval of the hazard ratio of 2 prior to approval, and then a hazard ratio of 1.4 at study completion, and that could be potentially post-approval. And so the question that we wanted to ask here was, just to describe some of the different phases of drug trials. If you want to just write some comments, you can choose one or all comments in the chat, because I don't think we have this one in the poll. So I'm just monitoring the poll. I'll give it a couple more seconds and then I'll move on if no one participates. So this one was actually a phase 3B, but pretty similar to a phase 4. So I guess I'll just move on to describing the study, the different phases here. And this is really just a review mainly for the first years. But phase 1 studies are typically pretty small. So 20 to 100 healthy volunteers are typically involved. It'll last several months. And then the purpose is really to assess the safety and dosage. And about 70% of those drugs move on to phase 2. Phase 2 is typically a little bit larger, so several hundred people, usually with the disease or condition. It may last a little longer, so several months to up to 2 years. And these are really geared towards establishing or generating preliminary evidence on the efficacy and side effects. Phase 3 are therapeutic confirmatory trials. So these are larger scale trials, usually involving a couple hundred to a couple of thousand volunteers who have the disease. These are like multi-center randomized controlled trials, and they typically last longer, so 1 to a couple of years, really aimed at determining again, kind of generating further evidence on the efficacy and monitoring for adverse reactions. This was actually, there was a lot of conflicting information in looking up the difference between a phase 3A and a phase 3B. I'm just seeing someone in the chat said, does 3B mean after NDA submission, but before the official approval? To address for additional concern. Yeah, so I think that's exactly right. So phase 3A is definitely pre-approval. And then phase 3B may happen after, it might also be pre-approval, but it may happen after initial approval and just before marketing approval. So just when I think one of the common areas where a phase 3B might be required is just to establish these more longer term endpoints, safety endpoints, sorry. Any questions about that, or did any of the faculty want to provide any clarification? If not, I'll keep going. Perfect. And then the phase 4 is really post-approval. So this is it's very clear and straightforward, and it's typically to address any unanswered questions.
It is.
Okay, so in terms of the study design of the LIGHT trial, this was a phase 3B randomized multi-center placebo-controlled double-blind non-inferiority trial. There was a lead-in period to identify participants or patients who didn't tolerate the medication or who exhibited poor adherence. One of the features of this that we found interesting was that the lead-in period was actually blind, like they used a blinded crossover design. So they had participants randomly assigned in a 1-to-1 ratio between the two sequences. So they either received 1 week of the active medication followed by 1 week of placebo, or the other way around. And then subsequently eligible participants were randomized to in a 1-to-1 ratio to either of the intervention or control arms. And then in terms of randomization, they used an interactive voice recognition system using block sizes of 4 with no stratification factors. And so one of our questions here is, what was the purpose of the lead-in period in this trial?
Any other participants? Yeah, we have a few more coming in. Short answers are great. All right, let's look at some of the responses.
I'm not seeing any answers here.
I'm sharing.
Do you see any answers on your end, Dr. Irvin? I just see 100% answered.
Okay, let me work on this. Let me see if I can get that share. You want to keep going, and you can use the chat.
Good. Yeah, that sounds good. So there's a question in the chat here. If the focus is on formal assessment of a safety signal, does the lead-in period not make our assessment of safety problematic? So I think my understanding was that the lead-in period was really to identify patients who weren't tolerating the medication and would potentially therefore not be adherent to the medication. I don't know if Lijuan or Dr. Irvin, you have anything to add to that. I think when we talk about these larger safety outcomes, it's different and sort of on a larger scale than just tolerating a medication or not. That was my understanding.
Yeah, I mean, it smacks a little. I haven't looked at this study carefully. But it smacks a little. I know it's in bup is one of the 2 active ingredients, and certainly in the case of opioids, there have been lots of these EERW trials, but that's not what's going on here. But those are enhanced enrichment randomized withdrawal. And setting aside the fact that you're withdrawing people from treatment, they, start people on treatment and then only keep the people that don't have toxicities and that are getting a treatment effect, which isn't, it's obviously a curious compromise or trade-off between internal and external validity. And so here, again, it's a different study design being used here, but I think that lead-in period is, you just have to take that into account, and we have to look at the numbers carefully as we're seeing how it changes the population that's ultimately being studied if you're requiring people to tolerate treatment and take the medicine and yada yada. So I just think we should, like, come back to this point down the road a bit.
Yeah, that sounds great. That's great input. Thank you so much. And our second question here was just about their description of randomization. And so for the people who, I guess this was also kind of geared towards our first year students, but anyone can answer. Was there anything missing from the description of randomization? And if so, what do you think was missing, or what could have been added to make it more comprehensive?
Please feel free to use the chat or unmute yourself.
I hope you'll tolerate my slow brain, but I was just thinking about what Caleb said, and I'm wondering, is their strategy so bad when they're looking for basically toxicity of the drug is the objective, right? It was non-inferiority for a MACE outcome. And maybe I'm being, but maybe that's the population you want to look at it in, is the people who are going to keep taking it to see if there's a long-term bad effect.
Right. Yeah, I think that makes a lot of sense. So just to address that randomization question, I think it was great that they sort of described the system that they used, but there are still, I think, a couple of unanswered questions related to who was implementing or who was using the system, and they definitely didn't touch on concealment of random allocation. So those were two things that I was thinking would have been nice to have been included. So I think we'll move on to the next slide. So the participants in this study were overweight or obese patients at increased risk of adverse cardiovascular outcomes. So more specifically, their eligibility criteria was adult women and men. So for women, the age cutoff was 50, and for men, 45 years or older, with a BMI of 27 to 50, waist circumference of 88 in women and 102 in men. And then this was restricted to patients with characteristics associated with an increased risk of cardiovascular outcomes, adverse cardiovascular outcomes. And so they had pre-specified categories for this. And this study included 266 US centers, so fairly large scale, over June 2012 to January 2013. So after that lead-in period, participants were randomized to either the intervention arm or the control arm in a 1-to-1 ratio. The intervention arm consisted of an internet-based weight management program with the extended release fixed dose of naltrexone and bupropion. And then the control group also consisted of the internet-based weight management program with a placebo. So this was more of an enhanced standard of care pathway, I would say. And then the internet-based weight management program included educational resources on healthy eating, exercise, and behavioral modifications. It also included weekly online lessons and weekly emails with lifestyle tips and reminders. And then they also had access to a weight loss coach, programs to track weight, meals, and physical activity, and then a meal plan. And so one of the questions we had here was, why do you think the internet-based weight management program was included for the control group?
So I think there might be ethical concerns if the control group just don't receive any intervention. And also this may help us to improve the enrollment because regardless which group that the participants will be assigned to, they will receive some kind of intervention.
That's great. Yeah, that's what we were thinking about too, is just given, you know, the benefits of a weight management program, it's probably not ethical to sort of withhold that from members of the control group. And then if you're really just testing the added benefit of the medication, then both the control and intervention arms should receive that weight management program. So that's great. Thank you, Xiangji. And then
There's one
Elizabeth.
question in the chat. Sorry, Olivia, to cut from
Yeah,
cutting
no
you off.
problem. Elizabeth is asking about the difference in age between men and women and whether this might affect the generalizability of the intervention. And I think they didn't really touch on why they had different cutoffs for men and women. I would assume it would be related to cardiovascular risk profiles. But I'm not 100% sure. I don't know if Lijuan or Dr. Irvin, if you have any comments on that.
I would think that is actually probably the case, not knowing exactly as well.
Wonderful. Okay, if there are no other comments, we can continue to talk about outcomes. So the primary outcome in this study was the time from randomization to first confirmed occurrence of MACE. And then the secondary outcomes were times to first occurrence of MACE or hospitalization for unstable angina, fatal or non-fatal stroke, fatal or non-fatal myocardial infarction. And they also looked at changes in body weight, BMI, and waist circumference. And then most of these outcomes were adjudicated by a blinded independent clinical events committee at Cleveland Clinic. So in terms of the statistical analysis, I'll do my best. I found it very confusing and a little bit hard to understand really what they did and why. But luckily, Lijuan and Dr. Irvin walked me through it yesterday. So for the primary endpoint, they had 3 null hypotheses that were pre-specified. And they're basically based on different hazards ratios. So the first is that the hazards ratio for naltrexone bupropion relative to placebo is 2 or greater. The second is that the hazards ratio is 1.4 or greater. And then the last one is really the test for superiority with a hazards ratio of 1 or greater. And then the hazards ratios and associated confidence intervals were obtained using Cox proportional hazards model. And they also use Kaplan-Meier method to plot time to events for the associated outcomes. And so what's really unique about this trial is they used sequential testing procedures to control for a type 1 error rate with an alpha of 0.025. And so basically it was kind of like a stepwise approach where initially they had an initial non-inferiority analysis that was planned after accrual of approximately 25% of events. So that was 87%. And their plan was to start with an interim analysis to rule out a non-inferiority margin of 2. So that would be testing the first null hypothesis with plans to stop the trial if the non-inferiority margin of 2 is not met. If it was met, then the trial would proceed to at least 378 events to rule out the non-inferiority margin of 1.4 at the end of the study. And this was to address the second null hypothesis. And then finally, if the second hypothesis was successfully rejected, then they would proceed to test the third hypothesis, which was the test for superiority with a hazards ratio of 1. And then just a little bit more detail on the interim analysis.
I'm sorry, I skipped the previous slides. This is what Olivia was reviewing for 3 hypotheses. All right, moving on to interim analysis.
Thank you. And this is just a little bit more detail on the first interim analysis was planned at 87 events or 25% of events to assess that non-inferiority hazards ratio of 2 for consideration of regulatory approval. And the interim monitoring boundaries allowed for early termination for either superior efficacy or established harm, but not based on the non-inferiority considerations. And so before we get to results, we just wanted to ask a question. Hopefully we'll get some participation here. So which of the following outcomes, so going through outcomes A to E, which of them show that the test treatment is non-inferior to the active control? So really looking at that margin that says 1.2, that divides, and that kind of helps us to understand which might be non-inferior. So Elizabeth is saying all of them. Any other answers?
I would probably say ABC. B is probably like a typical situation. For A, it's not only non-inferior but also superior as compared to the no one. And for C, it's a little bit tricky. So we can see that the upper 95% confidence limit is lower than the margin, which means the intervention is non-inferior to the active control, but its lower bound is also above the no. So yeah, so this one is a little bit tricky.
Awesome, thank you so much. So yeah, so that's exactly right. So we said ABC for non-inferiority. And then as was pointed out, A would also be superior or show superiority. Great. So I'll let Lijuan carry on with the results.
I would just say, I mean, there's always a margin, right? So I just think that's sort of the second part of the sentence here. So, I mean, when you're describing non-inferiority, of course, there's a lot of statistical assumptions or sort of stipulations around that. But I think being clear on that margin is important because that's everything in determining which of these, you know, make it across the line, so to speak.
Right. Great point. Thank you.
Yeah, and also to add on to that, if you have multiple looks into interim data, then you probably want to control the type 1 error rate properly so that to avoid multiplicity issues. So as we will touch on later, or recall from the earlier slides, that at different sequential testing steps, then you have to use slightly different alpha rate, and yeah, and this is something that we'll also type back later when we get to the result session. All right, so here is the console diagram, and as we mentioned, Olivia mentioned earlier, show the leading period, and here we see about 1/3 of people who failed, who didn't complete the leading period, are due to adverse events, and about another 1/3 didn't meet the eligibility criteria, and some also withdraw consent, or a couple of them was due to sponsors' decisions. Not sure what they were. And then after they complete the leading period, still a couple of them withdrew consent, ending up with 18, 9, 10 people randomized. So here I have a question for you. Which of the following is the number of the intention to treat analysis population in the study? Is it the 4454 in the placebo versus 4456 in the active, or the second one, the second row number?
I'm going to try to launch the poll and see if it works.
Could we look at the poll results?
Can you see the results?
Yep. Okay, so we have about half. Okay, so yes, the general ITT definition is just whoever will randomize to each of the treatment group, then ideally it should be the number one. But in this case, in this study, they define the ITT population as those who were randomized and also dispensed with study medications. So in all the subsequent analysis, when they refer to ITT population, they are using the 4450 in placebo versus 4455 in the active. Just to make a note here that when you review the data, when they say ITT, we also need to pay attention of their definition of ITT. So
That's
it's really-
Lijuan, you're not allowed to make up your own definitions.
Well,
In
you-
general, you know, if I start calling everything that's blue red, maybe that's a bad. So I would just say that they didn't follow the definition for ITT. Maybe they labeled it something different, but you can't.
Yeah, this is strictly speaking not an ITT population, the number two. Yeah, but that's what they call in the protocol. So when we review the results, we need to pay careful attention to their definitions as well.
Maybe they should call their ITT modified ITT, but I know that term is also often criticized.
Yeah.
It would be better.
Yeah, it would.
Yeah, it would be better, right?
And I would add, I'm surprised that they were able to put this into JAMA, calling it ITT.
Yeah, Ian.
Yeah, Lichuan, I guess everyone else. I was having trouble with the 105 who withdrew consent. Did they tell us somewhere, maybe in protocol or appendix or somewhere else about those? What happened to those? Or what were the reasons for withdrawal?
Yeah, I'm not sure.
Yeah, I don't know. I'm not sure. As well, there are several that we noticed that were excluded based on the sponsor's decision, and that was not explained as well. You see it at more than one stage, I think. Oh yeah, during the lead-in period for sure. So I don't think that was explained, to be honest. I'll look again.
Thanks, sir.
Okay, moving on to the baseline characteristics. So overall, it's balanced between the treatment groups. And I just want to highlight that in this study, over 80% are white. And also to look at the blood pressure level, we can tell that this is the population that have pretty well controlled cardiovascular status. And also from the previous eligibility criteria, people with more severe cardiovascular disease or uncontrolled hypertension are were excluded from the study, were not eligible for the study. So that may also affect how we interpret the study results and generalize the study results to our target population. So here is the participant flow after randomization. I just want to highlight that here in this study design, they do allow participants to discontinue treatment after week 16 assessments if they didn't lose more than 2% of their initial weight. And this was an approach selected by the FDA, the sponsor, and the investigators to reflect the use of anti-obesity drug in practice. And because that in practice, the patients and doctors won't continue the treatment of an obesity treatment if there's no weight loss. But that also will affect the treatment adherence and also because this is a long-term study, the retention of this population that we will see in a later slide. Right, so here I'm showing the results of cumulative incidence of study drug discontinuation. As you can see here, the first 2 weeks, the adherence were pretty good. But after week 16, only 72% in the placebo versus 64% in the active are still taking the medications. And after the first year, only about 26% versus 37% in the placebo and active, respectively, are still on treatment, and even lower after 2 years. However, I want to note that discontinuing treatment doesn't mean that they are discontinued from the study, from the follow-up. A lot of these participants are still being observed in the study even after their treatment discontinuation for observing their cardiovascular outcomes of interest.
That's horrible, isn't it?
Yeah.
I mean, did I miss something, or are we happy with those numbers? I mean, I thought they had a lead-in period to, select out people that were non-adherent.
Exactly.
So, I mean, these look almost worse than real world data. Like we typically say 50% non-adherence at a year for many chronic conditions. So, but maybe this was able to be anticipated based on the adverse effect profile of the drug or something?
I think weight loss trials are one of those trials that have really poor retention and adherence as compared to other kind of trials.
I mean, I'm not surprised to hear that there'd be really low adherence in the real world. I'm just surprised that in a trial that you wouldn't see much higher rates. But fair point. Like, we're not talking about treatment for multiple sclerosis here. So fair point.
Mm-hmm. Yeah, so before we move on to the look at the actual efficacy endpoints, I just want to show kind of a timeline here. So the original final analysis was planned when the study reached 375 adjudicated MACE outcome. And after the first interim analysis testing hypothesis 1, the DMC could consider stopping for overwhelming efficacy of overwhelming evidence of efficacy or harm at the 50 or 75% interim analysis. And the study design did not allow early termination for demonstration of non-inferiority. So in the subsequent slides, we will see results from the first interim, second interim, and also the final end of the study results at 65% information time because they terminate the study early. So here is the result from the 50% interim analysis. The hazard ratio of 0.88 with a 99.7 confidence interval ranging from 0.57 to 1.34. And we can interpret this as the weighted average hazard or risk of MACE outcome during the study period in the active group is 12% lower than those in the placebo group. And so for the composite outcome, it's really important that we also examine the individual components of the composite endpoints. Here we can see that the favoring of the active is mostly from the cardiovascular death, and the other non-fatal outcomes, stroke and MI, were pretty similar between the groups. And also want to note that there's not much difference in all-cause mortality. And I have a snapshot of the treatment discontinuation figure up right here to remind us that a lot of people actually discontinue study drug early. So in the primary ITT analysis, even though there are follow events, but when the events occurred, a lot of them are probably no longer on treatment. And they also show the end of the study 65% information time results. So with the more accumulated data, the results are showing less favorable than the 50% interim analysis. The hazard ratio is 0.95 versus earlier 10 points, 0.88. So now let's look back at the 25% interim analysis. So the upper bound of the hazard ratio for MACE is 0.9, clearly ruling out the non-inferiority margin of 2.0, supporting the FDA approval. Then let's compare results from all 3 time points. This should be a straightforward question. Which of the analyses show the most favorable cardiovascular outcome results for the active drug? Just looking at the hazard ratios. And also think about what can we learn from these results.
We have 8 responses. I'll wait just a moment.
Mm-hmm. Yes, correct. So with a hazard ratio of 0.59 at the first interim, it seems that it's favoring the active drug. However, it's been well recognized from a lot of clinical trials that early results can be quite unstable and may change throughout the course of the trial. And for this study particular, if the study had pre-specified the possibility of stopping for overwhelming efficacy at the first interim, then they would use a far stricter boundaries than the upper bound less than 1 in order to declare superiority. But they didn't have this in their study design. So using the result from the first interim to declare superiority is not allowed with the study design. So they also have another analysis population for efficacy outcome, which is the on-treatment analysis. Can anyone tell us who were included in the on-treatment analysis population? So what's the definition for the on-treatment analysis population?
I know the students may have some ideas because we sent them the questions, a little hint ahead of time.
Any takers? And also to think about why were there fewer events in the on-treatment analysis versus the ITT? All right, in interest of time, I will just give the answer here. So the treatment population, according to their definitions, the ITT participants who took at least one dose of study drug, but then they were censored after 30 days after the last study drug. So here I have a simple illustration of two participants. So participant ID 1 had a MACE event around week 20, and participant 2 did not have the event of interest, remain on the study for follow-up. So both of the participants discontinued treatment early. Here, the second panel here, around like week 10 and 12. So under ITT analysis, we will include event around week 20 for analysis. However, under treatment analysis, we will censor this participant 30 days after their last dose. The event observed won't be counted in the on-treatment analysis. That's why we are observing fewer events in the on-treatment analysis versus ITT analysis. So we only have about 35% of events, included 35% of events in the ITT population for the on-treatment analysis. And this on-treatment results shows less favorable than ITT. And also look at the cardiovascular outcome. So this is the most extreme difference compared to the two analysis population. So the additional 12 cardiovascular deaths in the placebo groups versus the one additional death in the active, favoring the active drug. They are all happening after participants are no longer taking the treatments. So just a quick summary to compare to analysis population. Why we prefer use ITT analysis population, because it preserves the integrity of randomization and provides a conservative estimate of treatment difference or efficacy. However, in the safety study, when the adherence is low, ITT can result in false positive declaration of non-inferiority. Versus on treatment analysis, it reflects outcomes while participants are still receiving treatments and reduce bias towards no difference introduced by non-compliance or protocol deviation. And in non-inferiority trial, it can help reduce the risk of falsely declaring non-inferiority.
I'm sorry, not to interrupt, but we have about 8 minutes left, just so that you know how much we have left to cover.
Yes. Yep. So quickly, the adverse events, we see significantly higher AEs in the GI, central nervous system, and psychiatric disorders, and the imbalance in the safety profile can pose risks of potentially unmasking the treatment. So, as you read the paper, you probably notice that. So they used the first interim analysis results and to support their approval, which FDA granted in September 2014. But the FDA stated that due to the breach of confidentiality, the trial could not be used to meet the post-approval requirement to rule out the non-inferiority margin of 1.4. But FDA encouraged the study to continue as planned. So why do you think the FDA rejected using data from this trial for the final evaluation? And why is the early release of the interim data problematic? All right, in interest of time, I'll just summarize here. So the risk of releasing interim data results has a potential to misinterpret the study results. As we've seen earlier, like how unstable the interim analysis results may be, and particularly in this study, so the interim analysis was not designed to test for superiority as for the first interim. However, it was clearly misinterpreted even by the sponsor, as we will see later. And it also has the potential to compromise the conduct of the study. Early knowledge of interim results may affect the behaviors of the sponsors, investigators, and participants in the trial, as well as actions of physicians or patients in the community. So recruitment or retention of the trial participants may be jeopardized. So if the interim analysis results are perceived to be negative, then participants in the study may incline to drop out or stop taking their medications. But if the interim analyses are perceived to be positive, then when the medication is actually available on market, then the physicians may opt out and put the patients on the medication, use medication instead. So all these behaviors can jeopardize the integrity of the study results. So at the so here I want to show that they actually for the confidentiality protection, they actually have a data access plan. So this is a document that outlines how data will be shared or made accessible during the course of the study or even after. And this is a document that strictly specifies who has access, and typically only a very limited number of sponsor representatives who can prepare reports for regulatory authorities and who agree not to reveal the data to other internal, external sponsors can have access. And so within the sponsor, they usually have a firewalls to separate out those blinded and unblinded groups so that they cannot share the confidential information. And this document will also typically have some confidentiality training or specifications. So for this study, in 2013, the sponsor presented the plan to the DMC, but the committee rejected their initial plan because with the statement that the business interests do not provide sufficient justification for gaining access to the confidential information or safety or efficacy. But then the sponsor revised the plan and make it to limited to individuals needed to get access. However, in 2 months later, they were still found to have over 100 people have access to the interim analysis results. And even worse, in March 2015, in the filing to SEC, Security and Exchange Commission, and even they published on their website stating that the drug had a positive effect on cardiovascular outcomes based on the results from the first interim analysis. And they have statement that the outcomes were expected and appear to unrelated to weight change. But ironically, on the same date, they also have the results from the 50% information time, which disagree with the statement that they put out there. And so the investigators of the trial recommended stopping the trial and release the 50% interim analysis results. Because they think that at this point, the integrity of the trial will be compromised, and the executive committee released the 50% interim analysis data because they felt that they had a duty to the public and also to the investment community to tell the truth. And this is something that is pretty unprecedented of committees to come out and release the data. So just a quick follow-up of where the cardiovascular results are at this point. After the termination of the LIGHT trial, they were required to conduct another study, which they initiated the CONVENE trial. However, it was also terminated because of some company merger acquisition transition. And the original sponsor of the drug, Orexig, filed for bankruptcy and was purchased by other companies that's currently the owner of this the approved drug. And with all this, the effect of Contrave, this is the brand name of the drug, on the cardiovascular mobility and mortality still remain unestablished. And this is the label from their website. We are kind of running out of time. This is a very interesting trial, very interesting case. And there's a lot of information to share. So does anyone have some final remarks or comments?
Any other questions? Any questions? Well, thank you both for an excellent presentation. There's so much to talk about with this trial and all of the other follow-up, and more than we can cover in this hour, but you've both done a great job. Thank you all for attending.
Thank you.